RTKs and kinase cascades. Receptor tyrosine kinases. Receptor tyrosine kinases, or RTK, transduce cell signals across the cell membrane. A kinase is an enzyme that transfers a phosphate group from ATP to a target molecule. The introduction of the phosphate group causes the target to change shape and change activity. So if we have a protein, we have our ATP molecule. The work of the protein kinase is to phosphorylate the protein by taking one of the phosphate groups from the ATP, resulting in ADP. RTKs have an extracellular ligand binding domain. You can see extracellular ligand binding, a transmembrane domain, and an intracellular kinase domain. The kinase domain can be phosphorylated itself on tyrosine, which is shown as the Y, amino acids. There are 3 steps to the process by which RTKs transduce cell signals across the membrane. Step 1, growth factor or hormone molecules bind to individual RTKs, inducing a conformational change across the membrane. Step 2, once the signal molecules are at the binding site, RTKs dimerize or pair up. So I have 2 RTKs here. The kinase domain of one receptor phosphorylates its neighbor on 3 tyrosines simultaneously. And then in step 3, the fully phosphorylated dimer is now active and transduces the growth signal to targets in the cytoplasm. So once we have our phosphorylated tyrosine, we can induce our cellular response. 58 receptor tyrosine kinases have been identified in humans. The ligands of receptor tyrosine kinases are often growth factors, molecules that signal the cell to reproduce. RTK are often mutated in cancer cells, where they transduce constant growth signals to the nucleus, even in the absence of growth factors. RTK mutations are common in tumors because they are one factor that keeps cancer cells multiplying when they shouldn't. Many modern cancer treatments are molecules that inhibit RTK or mutant RTK. Memorization of individual steps of signal transduction pathways is not necessary. Your goal is to become familiar enough with a few common pathways that you could identify, interpret, explain, or make predictions about a signaling pathway if you were given a diagram or description. Cytoplasm transduction. The cytoplasmic response is self-regulating. Its products will initiate negative feedback loops that reduce signal transduction. Negative feedback loops are crucial for cell survival or for the survival of the organism. Constant on signals are destructive and dangerous because they upset homeostasis. Signaling pathways that are constantly on or constantly off contribute to the development of cancer. Kinase cascades. Kinase cascades, or phosphorylation cascades, transduce signals in the cytoplasm. Cascades are carried out by a series of cytoplasmic proteins which amplify cellular responses. The general scheme is as follows. Inactive kinase number 1 is activated by a relay molecule, such as CAMP or a G protein, which we've seen in a previous section. Each activated kinase number 1 activates many copies of its target, kinase number 2, through phosphorylation. Each active kinase number 2 phosphorylates many of its targets, kinase number 3, and so on. The signal is amplified and terminated almost immediately, resulting in a rapid, powerful on-off switch that blasts a limited signal. One target of the kinase cascade will be a protein phosphatase, an enzyme that removes the phosphate groups from active enzymes, turning off the cascade. This shows the cascade with the flow of phosphate groups and energy at each stage. In this case, the two enzymatic products are two other kinases, named here kinase 2 and kinase 3. The chart shows an illustrative tenfold increase for each step. So if we have our relay molecule, our signal relay molecule, maybe it's CAMP, we take an inactive kinase and an ATP molecule, we get a phosphorylated active kinase 1. Inactive kinase 2, with the help of an ATP molecule, can then phosphorylate, and we have active kinase 2. And then again for active kinase 3, and ultimately we get a cellular response. And let's say that this can happen at each step 10 times. So ultimately we get amplification times 1,000. Kinase cascades are self-limiting. An enzyme activated early in the pathway will also phosphorylate a phosphatase. So that's what we see here. We have this negative feedback loop initiated in the first step here to give us an active phosphatase. Phosphatase enzymes work by removing phosphates from enzymes, inactivating them. So in this case, the phosphatase is activated in the first step so that active kinase 2 can be deactivated by the active phosphatase, and that breaks the chain here so that the cellular response ends. So we never make it to this third step in the cascade. Mitogenic pathways. A mitogen is a substance which causes a change in the cell cycle, often by inducing mitosis. A mitogenic pathway begins with a ligand and ends with a response by genetic material within the nucleus. The pathway includes a cascade with the activation of each of the following as shown in the table. The left column is the general name for the molecules along the pathway. So MAPKKK, MAPKK, MAPK. The right column contains specific examples of each from one pathway. So maybe RAF, MEK, ERK. The pathway is illustrated on the next two slides. This cascade begins with RAS here being activated by the addition of GTP and ends with an active transcription factor in the nucleus. So here's the nuclear membrane. This is in the nucleus. Observe the energy flow and roughly tenfold multiplication at each stage. So what happens here is we have this cascade where GTP becomes GDP in the process of phosphorylating this MAP KKK. That is amplified 10 times and continues the cascade MAP KK through the ATP to ADP process is phosphorylated to MAP KK. That's amplified another 10 times, so we have 100. Then the next step in the cascade is this MAPK. Again, is phosphorylated from an ATP to ADP. And then we get dimerization of this MAPK activated or phosphorylated. And that's amplified another 10 times. So we have times 1,000. And we get that passes into the nuclear membrane. We have an inactive transcription factor that once phosphorylated, again through an ATP to ADP conversion, is our active transcription factor. The orange bar at the top of the picture is the cell membrane. The bar at the bottom here is the nuclear membrane with a nuclear pore. This is the same cascade, but with the actual molecules, which are a subset of the types, MAPKKK, MAPKK, MAPK. So the MAPKKK is this RAS, MAPKK is MEK, and MAPK is ERK. The conversion of ATP to ADP provides energy and a phosphate group to the molecule, stimulating the next stage. Since these are kinases, hence enzymes, they are not consumed by the reaction and must be turned off to end the cascade. This is a drawing of a similar pathway which was used in the first question of the 2021 AP exam. While students were not expected to know this in advance, since it was explained in the question, being familiar with it would have been a big help. See if you can identify how this drawing relates to the previous slides. So first of all, you can see we have the plasma membrane, the cell membrane, and the nuclear envelope or nuclear membrane here. And you can see that this is targeting for transcription of target genes, so some sort of activated transcription factor. So that's just big picture. That makes it seem similar to what we saw before. But you can also see the cascade happening here. So this MEK to phosphorylated MEK, which stimulates the next step in the process, ERK to phosphorylated ERK. And the whole process is stimulated by this signal from the plasma membrane here, right? This signal relay molecule. We're not sure what it is because we don't have the description here, but whatever it is, this signal relay molecule is what kicks off the kinase cascade. How do I know it's a kinase cascade? Because what is the purpose of a kinase? It's an enzyme that phosphorylates, and I can see that we have MEK, phosphorylated MEK, and so on. Calcium ion flux. Calcium 2+ ions act as second messengers in many signaling pathways. When calcium ions flood the cytoplasm, they interact with protein targets, changing their folding and allosterically regulating their activity. Calcium ions are stored in the smooth endoplasmic reticulum or in interstitial fluid. One example of the action of the calcium ion flux, large release, is in muscle contraction. A flux of calcium ions into the cytoplasm of muscle cells activates myosin, a protein that must be activated for muscle contraction. Calcium ions are just one type of a second messenger in complex pathways that lead to many cellular responses. This particular example has 4 steps. Step 1, a ligand binds to either a GPCR or RTK receptor, which activates a membrane enzyme, PLC. So either the ligand binds to GPCR or RTK, activating this membrane enzyme, PLC. Step 2, PLC activates a second messenger, IP3, which you can see by this green hexagon. Step 3, IP3 receptors on the smooth ER open calcium ion channels, releasing calcium ions into the cytoplasm, which you see here. So there's a receptor that binds this IP3, signaling the release of calcium ions into the cytoplasm. Step 4, calcium ions in the cytoplasm impacts many protein targets. Fight or flight response. This is the response of mammals to a dangerous threat to their survival. When in danger, a rapid muscular response may be necessary. A mammal's nervous system causes adrenal glands to release epinephrine, a water-soluble peptide hormone, into the bloodstream. Epinephrine quickly reaches receptors in the liver, which induces signal transduction mediated by a GPCR and its downstream targets. One of the body's responses is the rapid release of glucose into the bloodstream. You can click the link on the video below to see a description of the epinephrine response.